Karyogram analysis. What is a karyogram? A karyogram is when you take a cell, a dividing cell, in most cases of a human being, and you look at the chromosomes and you can say something about the gender, that's the sex of the person. And you can also identify certain chromosome abnormalities such as trisomy 21 or Down syndrome. It's important to understand that albinism, Huntington's disorder, and these are mutations of individual genes and not of and do not result are not due to the change in the number of chromosomes. So you cannot use karyograms to identify, let's say, albinism. This does not work. There are now sex chromosomes and autosomes. I already mentioned this in a different video. Human male has 22 pairs of chromosomes, an X and Y. A human female has 23 pairs of chromosomes. The two X chromosomes are already one pair of the 23, and one pair is made of, again, of two homologous chromosomes. And each homologous chromosome is again made of two identical sister chromatids. A very brief drawing over here. Homologous chromosomes have the same size, have the centromere at the same location, and have the same genes. These two are sister chromatids, and these are referred to as homologous chromosomes. So for example, if you have over here eye color, then there must also be over here eye color on the homologous chromosome. So, but let's look at a karyogram and let's see what we can see over here. First of all, you can see that the chromosomes are arranged according to their shape and size. You can also see that the chromosomes have a particular coloring pattern, a banding pattern. This is because the scientists who extract these, if you stain the chromosomes, if you add certain chemicals, then they will give you this coloring pattern, which will help you in making a karyogram. And you see that these chromosomes all have a similar coloring pattern. Now, the question is now, are these now two sister chromatids or are they homologous? Well, these are homologous chromosomes. Why can you not see the sister chromatids? They are simply too close together, so that under the microscope it's not resolved. You have to look very carefully, but these are actually two sister chromatids sticking together, maybe held together over here, same over here, two sister chromatids over here, the same situation. This is a homologous chromosome over here, this is a homologous chromosome over here, and they're two sister chromatids very close together. The question is now, is this now a boy or a girl? And we have 22 pairs of chromosomes, and down here you can see an X and a Y chromosome. You see that the X chromosome is significantly bigger than the Y chromosome. It is not a pair. They do not have the same size, and therefore this is this is a male, and there are no chromosomal abnormalities visible. Maybe this person has another genetic condition. We cannot see that because this would be on a gene level. It's too small to see. But simply from the chromosome count, there are no abnormalities visible. The second example over here is a different picture. And here you can already see a little bit an indicator that there is there's two sister chromatids present. What about here, male or female? Let's have a look at the X and the Y chromosomes. What do we see over here? Well, what we have is an XXY. This is a chromosome abnormality. There's one chromosome too much. And this person is actually a male because of the presence of the Y chromosome. And yes, this is a chromosomal abnormality. It's called the Klinefelter syndrome. And there are a certain percentage of the population who have this, and there is one simply speaking, there is one chromosome too much. Yes, there is a chromosome abnormality here. So basically, a very common one where I do not have a karyogram right now. I'm going to quickly change colors here because on the it's a little bit difficult to see. I'm going to choose, let's say I'm going to choose black over here, is the following. Let's say that we have over here a third chromosome number 21, and let's forget about this one over here. Okay, let's say it's non-existent. In this case, no, this is not chromosome number 21, it's chromosome number 22. See? This is chromosome number 21 over here, right? It says 21. Okay. So, in this case, what we have is we have 3 times chromosome number 21. And this is called trisomy 21 or Down's syndrome. And this would be now a girl, XX, with trisomy 21, with Down syndrome. Are there other trisomies as well? Yes, there are. They're much more rare, but other trisomies are possible. I suppose the reason why trisomy 21 is more common than other trisomies is because if this happens, then there is at least still some chance that the embryo can survive. In other trisomies, I can imagine that if there is, let's say, a third chromosome number 1 present, there is so much DNA which is too much that maybe the cell has really a problem. The embryo, the cells of the embryos have a problem of dividing and surviving. So I think one of the reasons is that there's not a lot of DNA in chromosome number 21, and therefore it is possible for an embryo who has trisomy 21 to survive. How does this happen? Trisomy 21. Now I'll switch back to my white color over here. It happens if there is an egg cell with 2 times chromosome number 21 present. Let's not use this way of writing. Okay. 2 times chromosome number 21 present, and there's a sperm cell, and this sperm cell contains chromosome number 21 as well, and then essentially after fertilization you have chromosome 21 present 3 times. So let's go back. Important for you to know, is important for sex determination and for identifying chromosome abnormalities, but not useful for determining gene mutations in the DNA itself, because that is way too small to be seen and cannot be seen. The genes are way on a DNA level. This cannot be seen on the chromosome level.